Hello, welcome to Podovirus Podcast. We are back. Joe and I are here, and today we have Jonathan Solomon, who is the CEO of BiomX. So we are thrilled to talk to him because they listened to my wish I put out in the universe, and I said we need a positive phase 2 clinical trial for phages. Why hasn't anyone done that in decades? Actually, more than a decade. Jonathan said, okay, I'll do that for you. And then he agreed to come tell us about it because BiomX just finished a phase 2 clinical trial of diabetic foot ulcer. So this is also a clinical indication that I feel like the phage field has been always kind of seeing as like, why don't we just address that? Maybe that's the low-hanging fruit, or maybe it's really hard because it still hasn't happened, even though so many people agree that it would be an obvious indication because you can get the phages to the site and, they should work. And that's a very debilitating problem. So Jonathan's going to tell us how they did this and, what that means. And we have a bunch of our usual questions for him that we've been asking in our phage therapy series about kind of what the hurdles are for him these days as a pushing forward a company in the phage therapy space and what he sees for the future. So welcome, Jonathan, and maybe you can give us a little intro of how you came into phage. You had a life before phage. We can hear whatever you'd like to tell us.
Sure. So Jessica, Joe, thank you for having me. Quite excited. I've listened to your podcast and enjoyed it. And we know it's a small community, right? And it's great to kind of have this. And the more we talk to each other, the more we learn from each other, the better. And we're quite excited. I think this is quite an inflection point, and hopefully there'll be many more. So my background, a little bit on the unusual. I am Israeli. You can see from the hairstyle. I actually served 10 years in a classified unit, which I cannot talk about. Then came to Boston, got my MBA, launched my first company, which was related to phage, but not directly phage that kill bacteria. And after 10 years in the States, when our second son was born, my wife notified me that we are moving back to Israel. And she's also a classified unit, so you definitely don't want to mess with her. And then I was kind of like taking it easy, thinking what to do. Actually kind of fantasized about a PhD in phage and got to do some work at the lab of Weizmann Institute researcher Rotem Solek. And they were in the process of launching a small phage company together with Eran and Inab, two of the big names of the Weizmann Institute, as well as a funding from OrbiMed, which is a founding investor, together with Takeda and J&J. And I kind of started for a bit and said, hey, I really want to get my PhD. Maybe I'll be on the board. But you know how it is, kind of found myself a few months after that leading the company. So that's kind of how things started. We've evolved through the years. We kind of rode the wave of like excitement around microbiome and looked into IBD as well as acne. And then even went public, which was fun for a while, less fun afterwards. And we're fortunate to kind of build together a team that said, look, it's going to be really tough. Phage is a really tough modality. Let's kind of focus on an indication that at least we know there's no risk on the target, meaning that the target is validated. We know that there's challenges with the modality, right? The phage. And let's focus on cystic fibrosis. We focused everything on cystic fibrosis, fortunate to have 2 clinical studies that read out in February 2023 and then in November 2023, which actually showed really interesting activity. I think the most dramatic thing that we saw there was that 3 out of 21 patients on the phage arm actually got rid of their infection altogether. So that's what's called bacterial conversion. So that's dramatic. That does not happen spontaneously in chronic CF infections that are there for like 18 years. One of the patients was infected for 35 years, and after a few days of phage treatment, got rid of it. Right. So that was exciting. Again, it's not the clinical data that we now seen in the diabetic foot study, but in terms of microbiology, we saw a really interesting signal that got Deerfield excited together with the CFF Foundation, and kind of went together and put together a large financing. And as part of that round, we basically merged with Adaptive Phage, right? And that brought the DFO program. So much of the credit in the diabetic foot osteomyelitis program goes to the folks at Adaptive Phage and their thinking, right? We kind of the study was already like a year into enrollment, even more. And basically we took it, worked on it, kind of streamlined. There's a lot of work on, managing CROs, et cetera. But that's kind of the background, I think, to where we are today. Again, very excited with the diabetic foot program. CF study, we're about to launch the phase 2B. So hopefully kind of 2 shots on goal. That'll be interesting, but definitely the diabetic foot studies where we're seeing not only the microbiology effects that we've seen in the CF study, but actual clinical effect with statistical significance.
Amazing. Thank you. Before we dive into the diabetic foot trial, I feel like a lot of people have questions about the CF trial too. And so why do you think that there wasn't a clinical effect that you wanted to see with that one?
So here I think the answer is pretty straightforward. It's too short, right? I think it's and too few patients, right? So this is only 10 patients that we've dosed. We've dosed them, twice a day. Interestingly, we had an earlier small, like a 9-patient study beforehand, which is a pilot study. Again, everything is done hand in hand with the CF Foundation. There have been kind of the guiding light in how to go about it. Interestingly, and I think we should talk about also the diabetic foot study, a lot of the thinking was guided by compassionate use cases, right? So there is a lot of data out there, right? We could be skeptical about the data, but it still serves as guideline. And both the CF study as well as the DFO study kind of follow the paradigm that were set in the compassionate use cases, right? So in the CF study, we did a small study that was 9 patients that it started with like 1 day placebo, second day a low dose, third day a high dose, and then 4 consecutive days of a high dose twice a day. Right. And we knew from the compassionate use case that you need 10 days to really see a microbiology effect. So here we did see some reduction over like a log reduction between treatment and placebo. Interestingly, one of the patients that had a 3.3 log reduction, right? So 99.9% reduction of bacterial load, the physician called us a few months later and said, hey, this patient doesn't have the infection anymore. So this is like a patient that I've been treating for many years and boom, the bacteria is gone. Right. So that was encouraging. We launched the part 2. Again, it was 34 patients dosed for 10 days. Right. So it's not enough to see kind of the clinical effect that you want to see, meaning improvement in lung capacity or patient reported outcome. Not in terms of duration and not in terms of like the power of the study. Right. So I think we weren't expecting. We did see some signals, I would say, in FEV1 and in patient reported outcome. But again, I think it's such a small number that we shouldn't be hanging our hat on it. Some signal, interesting, until we do a larger study, and that's a phase 2B with 60 patients for 2 months, we can't say anything. But the microbiology, we did see this effect. I will say, by the way, another key important takeaway is that the background antibiotics were having a lot of effect, right? So in both our studies, DFO and CF, right, placebo is getting antibiotics and treatment is getting antibiotics plus phage. So we could see in CF, some of the patients have this, like they do 1 month on, 1 month off, or 1 month one type of antibiotic and 1 month another type of antibiotic, right? You would imagine that after like 10 years of this regimen, these fluctuations around the, changing the antibiotics would kind of stop. They don't, right? So I think that kind of created a lot of fluctuations that we saw. But again, we did see this kind of hard endpoint of like bacterial conversion, which is convincing. Again, trends in clinical endpoints, but I don't think it's sufficient to really say, hey, we're seeing something dramatic here.
For details, can you talk about what the primary endpoints were and how you decided on them?
In the CF study or the DFO study?
Yeah, and let's start with the CF study.
CF study was pretty straightforward. Again, it's only a phase 2A, so primary was safety and tolerability. We all know phage are safe, right? So that's a low-hanging fruit, but that was good to see. And then all the exploratory were microbiology, right? Whether we can reduce bacterial or see bacterial conversion, FEV1, which is basically the volume that the patient can kind of breathe, as well as what's called CFQR, which is a patient-reported outcome. And we did see trends. We did see trends in FEV1 and CFQR, which was great to see. I mean, in the 90s, when there wasn't a lot of antibiotic resistance, when you treated patients with antibiotics, you did see an effect within a couple of weeks, right? So it was encouraging. And again, I think the dramatic result that we saw in the bacterial conversion, which is what we got excited about.
Yeah. Just if I could ask another follow-up is,
sure.
In terms of exclusion-inclusion criteria, were you selecting the patients were included based on them having an infection of a what bug? And did you screen to make sure that the patients were sensitive to the phages you were using?
Yes to both. Yeah, yes to both. And I think that's crucial. And we'll probably talk more about it based both on the DFO and CF, right? So we made sure that the patients in the CF study was Pseudomonas aeruginosa, We took, sputum samples, we cultured them. We then made sure that they're sensitive to our phage, and those were the only patients that were enrolled on the study, right? Same in DFO, which made it even more challenging, right? We literally took bone biopsy and cultured the bacteria. And there was a major dropout because not all the centers know how to do a bone culture or a bone biopsy and grow bacteria out of it, right, successfully. So, as I think we think about, right, takeaways, we're very, very conservative in making sure that the patients had the bacteria and that the bacteria were sensitive to our phage. And I suspect that that's one of the key, reasons for success. I will also say one needs to be careful, even more so in the CF study, that there's high levels of bacteria, right? Because if you want to see a drop in the levels of bacteria, and we know that, CFU measurements are noisy, you want to make sure you have a high level of bacteria so you can see the drop, right? So that's also crucial. I suspect some of the studies that had low levels of bacteria, right, if they don't have the dynamic range, they just won't see the effect.
Yeah. Oh, there's so many questions. I guess first, just to finish up on
Fire
the
away.
CF trial. Yeah. Like, will you design? So the results that you got were positive enough that led you guys to decide to design a larger study. And do you change anything about the design in the larger study based on what you learned from that one?
So I think the most important thing is the duration, right? So I think what we saw in the phase 2, what we did is we tried to synchronize the phage treatment together with when they started a new antibiotic, right? It was only 10 days. So what we saw is that when you change an antibiotic, or, the patient kind of did not take an antibiotic for a month and then started taking an antibiotic for a month, right? There'd be like a few days that the bacterial levels are kind of fluctuating. And then after a while it stabilizes. And I think that's something that, if you dose long enough, right, you're not going to see that because you don't care, because it all averages out. And to be even extra cautious, we kind of made sure there's a run-in period. So the patients will start the antibiotics, first. A few days later, they would start the phage treatment. Interestingly, to that point, the patients, and this was fortunately pre-specified, the patients who were taking antibiotics, the same antibiotics all the time, we saw a 3-log reduction in the bacterial count, right? It's a very clean signal. So I think in those patients that are alternating or cycling, that's kind of the terminology, right? Whether they're taking, Toby and Strainum 1 month, or they're taking Toby for 1 month and then, antibiotic holiday for 1 month, those are the patients that got to be careful. And I think that's where you might ensure you dose long enough and there's a run-in period. So I think that's a key takeaway. Except for that, we want to make sure, again, we have those patients that are continuous antibiotics, and we make sure that there's high levels of bacteria to begin with. I think that's key. 2 key takeaways as we're trying to differentiate, right? Sort of asking, why did we see activity here and others have not? And again, compassionate use cases where the bacterial levels tend to be higher, they did see an effect, right? So it kind of holds together nicely.
So if I could ask another question, can you tell me what the route of administration was and how you decided upon that route
So
for
it
the
was
CF study?
nebulized. Yeah, it was nebulized based on compassionate use cases. And I think Jessica kind of mentioned early, route of delivery for phage is crucial, right? Because what we've learned, by the way, the hard way, in our early days, we had an acne study and we thought, hey, this is like a super low hanging fruit. Just tested. We took an initial clinical study that we saw sort of that the phage can actually reduce bacterial levels on the skin, right, with a skin swab. But the bacteria is actually inside the sebaceous gland. You got to penetrate really deep. And we had no ways of modeling it. There was no compassionate use cases kind of build that case, and it just didn't work. Right. So I think that was our takeaway, which is like, phage is a very large molecule. Delivery is not trivial. And if it dries on your skin, it probably doesn't sort of propagate. So you got to be very cautious. So I think our thinking is always like, better to be in a sort of liquid environment, be better based it on compassionate use cases, right? When there's some evidence that stuff works, right? IV, there's a lot of compassionate use cases that works. Topical in a wound is what we had in DFO, and nebulize, quite a lot of cases that show that it worked pretty well.
So did you how much did you experiment with different nebulizers, is I guess what I'm getting
Quite a
to?
lot. We built a whole setup in our labs, and worked in nebulizers and drop size and everything. And again, fortunately, there's good models for that, but we did all the work to make sure that the phage are intact and that the droplets are a size that penetrate deep into the lung structures. So we try to maximum that we can to kind of reduce the risk as we enter the clinical study.
It seems like those are some of the important things. And do you,
Right.
and
Go ahead.
out of curiosity, when you're picking nebulizers, is there any aspect of, I mean, how far down the road are you thinking? Are you thinking if this is the way we have to nebulize it, that it might be easier or harder for people to self-administer once the product hopefully is approved and in the market? Is that something that you're already thinking
It
about?
is.
Or
It is. It is because I think there's a precedent, right? These patients are taking Toby nebulized, right? So there's a mindset. They already know they want to do it not too long, right? There's a bunch of products on the market. So at least there's benchmarks and you're trying to optimize, of course, commercial availability, patient access, patient ease, how easy it is to work with a company that produces the nebulizers, right? And the performance of the nebulizer. So, it's a big matrix of parameters.
While we're on this subject of delivery, I think this is a really good segue to the diabetic foot trial, because I noticed you used IV in the first dose, and then the subsequent doses were not IV, but only topical, right? How did you get at
Right.
that setup?
So again, I think this is all credit to the folks at Adaptive Phage, and I think the rationale was to follow compassionate use cases, right? Adaptive was involved in some of the compassionate use cases, providing phage to that, and there's quite a few publications. And I think the rationale was like a bit like oncology, right? Like a single dose for like debulking whatever bacteria is sort of available from the IV route. And then topicals to trying to break down biofilm, and that would take a longer time to have the effect. So that's the rationale. I will say, interestingly, when you look at the literature, right, osteomyelitis kind of sticks out as one of these indications that there's more data in compassionate use cases than others, right? And you sort of say, why is that, right? Like, I think we all know, right? There's compassionate users that are successful and less successful. And again, the literature search that we've done, and obviously Adaptive has done at the time, osteomyelitis and diabetic foot kind of stick out as one of those indications that there's more efficacy. There's, for example, a paper coming out of Fish of Washington University of 11 out of 12 patients that are having like a very positive response in diabetic foot. And I think as we try to think about it, right, you can say, okay, there's poor blood supply, right? So antibiotics are not getting to the site of action. So maybe phage, because it amplifies, will have an advantage. B, a lot of antibiotic resistance, right? A lot of MRSA and some of the other nastier bugs. Okay, makes sense. Phage will be orthogonal. And 3, there's a ton of biofilm, a lot of biofilm in those indication, right? So again, I think it's these 3 aspects. Again, we can theorize, right? It's hard to measure all these things, but I think at least that gives the basis why you're seeing greater efficacy in DFL already in compassionate use cases. And again, our rationale, the folks at Adaptive, that's why we liked them and sort of bonded with them quite quickly, is to say, okay, there's a lot of data out there. Let's try to kind of integrate all the data out there in literature to improve the chance of success, right? We know it's already so tough, right? So let's kind of minimize the risk that as much as we can.
Yeah. And do you also think, so I think I want to talk about how you chose which phages. And I noticed, I think on one of your slideshows that you guys have on your website, that you are using, it's a personalized approach here where there's a bank of staph phages that each patient might get a different set of phages from that. Can
Right.
you talk about that
Right.
a little bit? And how big is your bank of those phages, things like that?
So APT has a bank of like 200 phage, right? Roughly less than a quarter is phage for Staph aureus, so it's quite a lot. The ideal is to use the platform, the PSD platform, to kind of optimize which phage to use together with, all the parameters, like what antibiotic the patient is taking, whether there's biofilm, try to optimize all the parameters and actually give a single phage to that patient, right? So it's kind of optimize all the parameters. It's a single phage. It says, okay, use this phage for this patient, right? So that's kind of the way it works. Yeah, trying to optimize, sensitivity, background antibiotics, biofilm, and whatever growth curve information we can produce.
Interesting. So, okay, so they only each get one phage. How many times do you feel like, or we probably know, or if you can say, how many times are the patients getting the same phage? Like, did you see certain phages pop up as just like the best choice most of the time?
Yeah, I will say I think we have experience already at BiomX with Staph aureus, right? Like I think Staph aureus, very much like acne, is one of these bacteria that you need fewer phage to kind of get coverage. So you do see like these phage that outperform the others, and I think that kind of informs as we think about the next steps. We have seen some phage which are again like this, like the C. acne bacteria. Probably like two phage give you like a 90% coverage of all different strains that we've seen, at least in our labs, versus like some of the others, which are much tougher, like Klebsiella, right? A lot of diversity, a lot of genomic diversity, meaning that it's much harder to come out with, a single cocktail that has broad host range. So I think it's a trade-off, right? I think there's some cases which lend themselves more to this personalized approach. Some cases will lend themselves to a cocktail approach.
So with a single phage, and of course this is obvious, but a lot of people get very worried about resistance. The bacteria become evolving resistance to the single phage. And one of the rationales for using a cocktail is that you have a chance that one of the other phages will kill the resistant one. So
Right.
how are you how concerned are you about resistance when you use a single phage, and what do you do to mitigate those concerns?
Great question, right? I think we have perspective from both aspects, right? So CF study, we have used a cocktail, right? We built a cocktail Pseudomonas. We treated, again, albeit it was much shorter. It was only 10 days, twice a day. We have not seen resistant evolve at all, right? So it's a cocktail on top of antibiotics. Here in this study, we have not seen resistant evolve either, right? Bear in mind, it's also on top of antibiotics, but I think we have to be careful in the way we measured it, just because we're taking a swab from the ulcer, which is not, unlike the screening, which is a bone biopsy, which is very clean, right? Toward the end of the study, we took swabs from the ulcer itself, which is a bit of a messier measurement. So we're not seeing resistant emerge. So that's kind of the data. Again, I think resistance on top of antibiotics is less likely. So currently the data, we've not seen it. Again, I think in the case of Staph aureus, we have the option to move kind of straightforward with a cocktail. Again, we'll finish to analyze all the information, but at least from what we've seen to date, we have not seen resistant emerge. And again, the personalized approach kind of allows you, if you see a resistant emerge, you can switch a phage if you want to.
Right. And if I could just follow up, did you look to see if just in a test tube you see resistance? Because there's these reports that when phages become resistant, when bacteria become resistant to phages, sometimes they are no longer as resistant to antibiotics, or they're no longer
Right.
virulent, and those are reasons why you might not see it. And so I guess the simple question is, do you see resistance in the test tube?
Of course, if you wait long enough, but in none of the cases was this, resistance sort of developed faster than with the antibiotics that were tested, right? So whatever it is, even the single phage is better than a given antibiotics from at least the data that we have.
Yeah. So on the zooming out to this trial versus other trials, I feel like, and maybe we can remind the audience, has there ever been a diabetic foot trial with phages that has been, controlled trial and actually gone to completion before this one?
Not that we're aware of. I mean, we've seen, I think there were some small ones. I think there were a few small ones from the European companies, but, it was like just a handful of patients that we've seen. And then quite a lot of compassionate use cases from what we were aware.
Do you have a sense of why that is? Like, why didn't anybody, if this is low hanging fruit, or maybe it's not, but why hasn't anybody tried to do that? Has it just been about raising the funding, getting all the ducks in a row, or any other reasons you can see?
I sense it, I sense that it just requires a lot of resources. Like I think this study has been going on for more than 2 years. As I said, like because we've insisted on taking bone biopsy and growing bacteria out of it, and making sure that the phage is sensitive to those in the bone. It's a very careful screening criteria, long, sort of long dosing, and quite a complicated regimen. My sense, right, again, I'm sure everyone kind of knows the inside story, right? But, I get the sense that a lot of it is just the financial resources you need to put into a study like that. Beyond that, we all know, right? Like we all have like our, phage lore, right? There's, what I call precision medicine, right? The fact that unlike antibiotics, you have to make sure that the patient has the bacteria. We know that, sometimes the bacteria kind of change seasonally in some of the infections, right? So making sure that it's not just a typical infection with a typical bacteria, but which bacteria are there, and you verify that your phage is sensitive to it, right? There's been a lot of issues on manufacturing through the years. I mean, we've raised north of $200 million, right? There is a lot of knowledge in the company that was learned, some of it in the hard way, right? So we all know, right? Like manufacturing is complicated, and you got to make sure that the titers are high. You got to make sure that they stay high. You got to make sure that there's no kind of infections from prophage, et cetera, et cetera, et cetera, right? So I think we all know of many studies that the material was just not of good quality, and you just lose like 2 logs in your titer, and that's it. You have no chance of seeing an effect, right? So so I think it's the GMP consideration, sort of it's a delicate biologic. And lastly, I think to Joe's point, like we've learned more about defense systems, and I think that's where a lot of the know-how that we bring to the table based on the work from the Weizmann Institute is actually understanding defense systems and which phage to use, right? CRISPR, right? That's the bacterial adaptive immune system, but there's so many other defense systems, and how you choose your cocktail and how you build in, which one you select. And if you're using individually, which phage you want to use, all that kind of goes into the picture. So, I think there's a lot of parameters that, again, I think we're all drinking our own Kool-Aid, but feels like, us and others are getting a handle, right? Like I think we understand how to run these studies properly, and hopefully we'll see more of this good data.
Yeah. Is there anything that you, oh, I have so many more questions. Anything you wish that you designed differently about the DFU trial that you're like, oh, if we only would have, collected this or done it slightly longer, or anything that you wish you'd done differently?
I think I think we all wanted to see a bit more microbiology data, right? But to the point that we talked about, it's really tough to be accurate in microbiology in this indication. So unlike CF, you measure the sputum before and after, and you just quantify, and sort of it seems to make sense. And the patients are adapting it. Here, I think the microbiology is an issue, right? So I think that's where we felt that's very risky, because we're going in blind, right? We can say if the study fails, we're not really going to know what happened, right? Like, for example, think about one of the questions and sort of bets that the company took is we also enrolled patients that had polymicrobial infections, right? And because all the experts said, this is like it's the head of the snake theory, which means that, right, if the patient has staph and some others, the head of the snake is staph, and if you take his staph, you should see the effect. And it worked, right? And I think we've learned a lot, right? Because think about it, this is like the first time we can actually test this hypothesis, right? Because we're taking out only staph.
Yeah.
But it could have failed. And then we'd be scratching our head and sort of saying, did it not have the effect, right? Like, did we reduce the bacteria? So I think I wish, and I think that's something that we're thinking, what can we do in order to just get better microbiology readouts that we have in CF, but here it's much, much more complicated.
On the line of polymicrobial, I've seen I think Technophage has a trial going on where they have a multi-species cocktail for diabetic foot. I think it has Pseudomonas and Acinetobacter phages in it too. But it sounds like the head of the snake theory panned out for you, and that you took out staph and it seemed to help a lot. Do you have a sense that there is a need to have phages that target these other pathogens as well?
Potentially might get better effects, right? We don't know, right? I'm sure it's beneficial to try to take out some of the other bacteria. Again, I think some of the big advantages with phage, right? This thing is safe. So if we can take out more bacteria, hopefully that's better. And again, now we have the tools to kind of test it, right? Here we're fortunate because the head of the snake theory panned out, right? By the way, it's kind of funny because we talked to all the KOLs. They're like, of course, we always knew it was true.
So
right?
interesting.
But,
yeah.
yeah.
Yeah, I've heard that from there was a phase in time where we went and interviewed a bunch of like diabetic foot doctors in various places. And yeah, they just had such a handle over at least like the mental model of what these infections are. And they're just like, yes, take out staph.
Right. But United, sometimes you kind of overthink these things, so you could be really worried because you could say, wait a minute. I remember that in our CF study, you can sometimes culture more than one strain of Pseudomonas, right? So does your phage cocktail have to be sensitive to, kind of all of them? And maybe we haven't captured all of them. Maybe we didn't grow all of them, right? And it's sort of like, you're like panicking before the clinical study. And in the end, just got to do the study and see what happens.
Yeah, I think that's, sometimes you do just have to, as they say, do the experiment.
I hate when they say that.
As a
I
Someone who
feel
worked
you.
on the bench next to me back
Yeah.
when I was doing bench scientists used to say, do the blanking experiment. But I mean, I guess, and that's true. That was in a yeast cell biology lab where you could do the effing experiment in a day or two, and you didn't have to enroll
Exactly.
patients and invest so much. I mean, I guess you still want to do the blanking experiment, but I guess the question is, and Jessica alluded it to a little bit, and I know that from hearing some of the other stuff that you've done, can you talk a little bit about the importance of talking to clinicians and seeing, I guess just sort of like when an antibiotic works, what do you reasonably see and in what time frame? Because it seems to me more and more that if you don't have those conversations, you're going to ask the phage to do something that even antibiotics don't do when the bug is sensitive. And
Completely.
that's, I think, in some cases been a huge problem for people who are trying to get the phages to do something that someone would never expect an antibiotic to do. And so I guess if you could talk a little bit about the need for that and how you've used discussions with people who deal with this condition all the time.
So I think you're hitting the nail on the head, right? It's sort of crucial because in the end, right, we're all a bunch of scientists and we're not the physicians. I'm more like a scientist wannabe, but let's put that aside. But the real people, the real scientists in the room, right? They're sort of like they're making the cocktail, they're thinking about all the stuff, but they're not treating patients, right? And it's very different because, again, think about like in our diabetic foot osteomyelitis studies, right? These patients are getting antibiotics. So we kind of talk to everyone, kind of say, it kind of goes to the question of dynamic range. Like, what can we expect to see? And the experts told us, look, they are being treated on antibiotics. So in 12 weeks, you should see a reduction of 40% in the size of the ulcer on the placebo group, right? Because, they're not like, it's a dire condition. By the way, it's horrendous. DFO, like 40% of the patients end up with an amputation, right? This is no joke. And the ulcer does shrink. So then you kind of say, okay, well, if placebo is shrinking by 40%, what do you guys want my phage treatment to be, right? Because is it 5%? Is it 10%? And they're like, no, it's got to be 20 to 30%, because if you don't do anything beyond that, then it's not really moving the needle, right? So I think those are the conversation that you want to have. And then they kind of suggested that we do, sort of like the tissue involvement, meaning putting a Q-tip and measuring how deep the ulcer is. Turned out to be super crucial, right? Because in our treatment, we saw an effect which was almost 40%, sometimes even greater than 40% with stat sig that floored us, right? Because we weren't expecting to see stat sig. And also, and this is kind of gory, but they put a Q-tip and they're trying to see how deep the ulcer is. So in the 13 patients with phage that they saw that they could literally with a Q-tip reach the bone, 12 of them had like significant kind of buildup of tissue over the bone after 12 weeks of treatment. And the placebo, only 5 out of 9, right? And some of the KOLs, by the way, to your point, kind of said that's actually more dramatic than the ulcer size, right? Because that's really when it heals and sort of like tissue is being built on it. By the way, again, right? Think about it. Like, I was like, wait a minute, 12 out of 13 patients. That reminds me the WashU paper, right? Which showed that, 11 out of 12 patients had recovery in DFO, right? So again, we're kind of repeating these compassionate use cases, and they kind of deemed that the KOLs were like, this is what we want to see. This is significant, right? So you got to be very mindful, because to the point that we said, like, if you're not having that 30% improvement, right? In the end, no one would pay for an expensive phage therapy if you're not having a dramatic effect, right? So you might be jumping up and down that you had a 10% improvement. Maybe it's stat sig, but no one really cares, right? Because it's not clinical meaningful. And again, I think we all know, right? We all carry scars and we know how difficult infectious disease are, right? And I think, right, again, we're trying to kind of pursue that path, right? I think some of them make more sense than the others for commercial reasons. And those are the reasons that we're trying to pursue. I think one company that I think has been our sort of like case study, or, the company we want to be, has been Insmed, right? Insmed went after a pulmonary infection called MAC, Mycobacterium avium complex. It's 12,000 patients in the US. They showed that after 4 months of treatment, the difference between placebo and treatment was like 20% bacterial conversion. So 20% of the patients kind of got rid of their infection. That got everyone super excited. That drug is generating like $300 million in sales, right? In anti-infectives, right? So, and we're like, wow, that's unbelievable. And those, by the way, in our CF study, we saw that after 10 days, we had a 14% conversion. That's where we said, yay, we're heading in that direction if we replicate in a larger study. But there are these cases that make sense. And that's where, the feedback from the KOL, the feedback from the payers, which is also crucial, right? And the science kind of all converge to deliver something that sort of, provides value for everyone.
Yeah. Amazing. One more thing on the delivery. Did you find with the topical that you had to get like way into the bone or like use a needle to get it really deep, or was it kind of in a bandage, or how did you actually deliver it topically?
It was a liquid administered over a bandage. I think, again, some of our takeaways from playing around with topical delivery, which is much harder, right? I think in an open wound, because the environment is humid and the phage is in liquid and you close it with a bandage, it's probably the reason that the phage can propagate and diffuse and you're getting a greater effect. Like, I think our conclusion that if you take a liquid, just put it on the skin, not in an open wound, just on a semi-healthy skin, it's probably not going to work. Right, because it's going to dry out immediately, and then the phage just won't be able to diffuse at all. So keep it in liquid, keep it very moist, and most of the wound is probably accessible from that point.
And do you have to care about, this is kind of like back to Joe's nebulizer question, but like, did you have to screen a bunch of different types of bandages that wouldn't stick to phages too much? Or was that really a concern of yours?
We did some work, but I think overall, it kind of, we try to be as much as we can with standard of care, right? Same with like nebulizers. You don't want any freaky nebulizer that they'll, need to get like a new house to contain, right? So we gotta back to consider the patients as well, and compliance, of course.
Yeah. And so looking forward for what's next for this? How do you build on this trial? You design a bigger trial or a different one, or what's next?
So one of the really interesting thing with the diabetic foot osteomyelitis study is that most of it is actually funded by the Defense Health Agency and the folks at the Navy. They put significant amount of capital into this, right? And one might ask why, right? And they're actually closely monitoring what's happening in the Ukraine with troops coming back with like super resistant infections. So they've been a big partner and supporter, very much like the CF Foundation was in a CF study. So obviously we're talking to them and kind of thinking about the next steps. But basically, it would be a larger study. I think there is a chance potentially to go for a pivotal study. This could be, an orphan indication. This quality of data could qualify potentially for breakthrough. So there's a lot of possibilities and flexibility to take it forward. But, again, right, finally, we have that phase 2 data. And I think we want to just move as quickly as we can forward and just take into consideration like the partners that are involved and have been a big supporter and funder of the program.
And this is, I'm just assuming this is the U.S. military, right?
Yes, it is.
Have you had any conversations with the Israeli military, or is that classified? Sorry, just kidding.
No. It's, again, I don't think they have the scale that the Navy can put into place, right? Unfortunately, we know drug development is so expensive that you need a very large scale. So, at least to my knowledge, the Israelis have not been doing at least biology on this scale. Definitely other stuff, right? But not biology in that scale. So I think the U.S. Navy and the Defense Health Agency is kind of our, go-to partner on this.
And when you do scale it up on the phage side, do you see the personalized one phage with a phagogram ahead of time being the approach even in the larger scales, or would you have to develop a cocktail based on the data to get it to the full scale that you'd want?
So I think we need to make the decision. I think in the case of Staph aureus, we have the flexibility, right? We have a lot of experience both with cocktail as well as the personalized. So I think we'll need to make that decision pretty quickly. In general, to your point, I think there's some indications that a cocktail will be the right way to go. And some of them, you have to be personalized, right? So I think Staph aureus, you have the flexibility. If we're talking about intimate, a bacteria like Mycobacterium, which, Graham Hartful has done some great work on, right? That's such a difficult bacteria. I can't believe you can develop a cocktail, right? So that's got to be personalized.
So maybe we should I'd like to go off of what you were saying about the funders. Obviously, the elephant in the room is that funding in the US government is shaking up a lot, especially with biomedical research, and infectious disease is sort of seeming on the chopping block. How are you thinking about that, and how is that playing out for you? Is it like as bad as it sounds like it is? I know we're all kind of watching the
Yeah. The answer is we don't know, right? We'll have to wait patiently and see. On the one hand, right, like defense tech is sort of like booming to some aspect, right? The fact that everyone's sort of like, building up their reservoirs and arming, etc., should be interesting because again, the Navy's sort of seeing what's happening in the Ukraine, right? It's kind of crazy, right? Troops are coming back with like very resistant infections. The war in the Ukraine looks like World War One. They're in the trenches and they're infected by everything in the mud. So I think there's a clear need. So I'm sure there's a path on that aspect. Again, I think it's been challenging, right? We've all had these issues. I think we've been fortunate to have great supporters, Deerfield, the CF Foundation, the AMR Action Fund, OrbiMed, Natanhala. But again, this has not been a walk in the park, right? This is not like with the companies that usually sort of fund big oncology studies. So I think it remains a challenge. On the one hand, right, we're sort of saying, you're seeing all this talk about antibiotic resistance and sort of the next pandemic, but we're not benefiting from the resources that we want to see. Right. So I definitely think it's a challenge. But we've been fortunate with the funders that we have, but this has not been easy. And, it's going to be more challenging going forward because it's going to require more capital, right? As we make progress, you need more money to do bigger studies. So again, it's good to get partners such as, non-dilutive partners such as the folks at the Department of Health, the Defense Health Agency. I hope that, what we all wish for, like getting positive phase 2 data opens up more BD opportunities as well. Like I think pharma will need to kind of enter the space. So let's hope we see that as well.
So obviously non-dilutive funding is preferred, but do you talk to venture capitalists, and are they feeling, one of sort of the naive thoughts is as the government pulls back, I guess, how does that affect venture capital's view as to when they think it's a good investment to get into this space?
Yeah, I don't have a good answer. I'm the entrepreneur, right? So I just knock on everyone's door and nag them to death and hope that they'll invest in hope of having to spend less time with me. But I will say some of the bankers that we talked to said that in the last 2 weeks, investors have been hiding under their desks. Right. So I think this is quite a challenging time. Let's see. Right. Let's see how we come out the next few weeks. We'll see how it is. It's not easy out there. Right. Like I think biotech had a very nasty run, which makes it extremely tough. I hope that with more advanced clinical data, it's easier. Right. And I think what we're trying to tell people, like with phage, you don't have the risk of safety, right? This is a safe modality. We're choosing targets which have been validated, right? We all know you want to take out Staph aureus and Pseudomonas. And hopefully the data that we've just generated in both studies is a major de-risking, right? So the risks are rather low for an unmet need. I think we're trying to talk to potential partners and saying, look, what's really interesting about phage is say you want to go after mycobacterium, right? You can literally, produce the phage cocktail, right, in GMP grade, hopefully within a year or so. And then you don't need to do animal studies. You don't need to do healthy volunteers. You go straight to phase 2, right? It's really attractive for a platform. Still a difficult, it's still a difficult sell. And again, that's why I'm crossing my fingers that more of the phage companies out there will provide, and deliver good data because it's good for everyone, right?
2 things you mentioned. 3. I will have to pick one. First, pharma. I feel like when I talk to people who work at pharma companies, I try to ask them, why isn't pharma investing in any antimicrobials anymore? I get why they're not, and we've had past podcast episodes diving into the pricing models and just like the economics of making an antimicrobial actually make enough money to cover its costs is hard. Do you think there's what would it take for pharma companies to actually be interested in what you guys are developing? Like, are you looking towards certain milestones that you've had conversations that actually they're not against it? it's just like certain things have to be hit that you're working toward? How do you think about all that?
So I think it's two things, right? Like I think and you can understand, right? The typical pharma executive will say, look, infectious disease is not that exciting. And phage is this new modality. I mean, it's not even a new modality, right? You guys have been talking for the last 100 years, right? Is there convincing data? Like, because some people would say it just doesn't work, right? People have been talking for such a long time, it just doesn't work. Let's move on.
Mm-hmm.
And I think hopefully now, right, we put a dent in that theory. So hopefully people will be like, maybe it does work, right? Maybe it's worth taking a risk. And then the second thing is the commercial potential. And I think that's, again, there are many indications that don't make sense commercially, right? Like why, if I can treat it with the standard antibiotics, or, only like in very few cases do I need like the fancy expensive antibiotics, you're not going to make any money. Right. So, and again, I think that's the unfortunate reality of the field we're in. But I think people like Insmed and others have cracked it, right? Like, you have the SMAC, right? Which is a really attractive indication, right? That's generating substantial amount of sales because of the patient population, the fact that they need it, the fact that the effect is so great, right? That it kind of makes sense. I think to kind of give you another example, bronchiectasis, right? So Insmed, again, nailed it with a study. This was an anti-inflammatory in bronchiectasis, by the way, an indication that no one touched for many, many years because everyone failed. And suddenly, they kind of designed the right study and they kind of nail it. And by the way, their market cap went from like 3 billion to 12, right? On that positive phase 3. And you think about bronchiectasis is very much like CF, right? It's just 10 times larger, right? Really interesting market that have bacterial infections, right? So there's an opportunity there. So I think it's got to be the combination of, pharma companies being more comfortable with the modality, meaning we generate more good data. And B, it's got to be a really interesting indication commercially because they've been burnt, right? But there are indications in the ID space that do make sense.
Yeah, I've heard about this Insmed before today, just once. And I feel like I heard about it in the context of, are they able to price their antibiotic against Mycobacterium differently than a typical course of antibiotics? Is it more of a like annual chronic sort of thing, or how does that work?
I know that they're charging, again, the dosing can be quite long, but they're dosing like $150,000 to my best of my knowledge, right? So they can command very high prices and payers are happy to pay that, right? Because if you've converted 20% of the patients, right, you've completely changed the outcome of this disease, right? Like from like, someone who just, is not being able to function to like a healthy person, right? That's really dramatic and they're willing to pay for it, right? So I think and it's a large chunk of patients. So I think indication selection, and I think it goes to Joe's point about it's the KOLs, it's the pharmas, and it's the payers, right? Like you got to talk to all of these different parties to understand and choose indication wisely.
So I guess my naive thought in terms of payers paying is that the diabetic foot indications where, the consequence can be amputation, which I think would be something that I would think naively an insurance company would go, we'll pay a lot to reduce the number of amputations among our patients. But
so that's our bet. I got to give the credit to the folks at Adaptive and Deerfield, our lead investor, for doing a lot of the work and choosing that indication. But yes, think about it. The numbers are insane. There are 160,000 lower limb amputations in patients with diabetes alone. 85% of them are driven by DFO, right? So just think about the number of amputations. As I said, there are like 200,000 patients in the US with DFO. Say half of them have Staph aureus. That's pretty much a number, slightly less than half. And if you have DFO, the patient will end up at 40% of the time with an amputation. By the way, the 5-year survival after an amputation is like 50%, right? That means it's worse than cancer. Like it's worse than the worst cancer. So it is a big unmet need with very large numbers. And just think about it, the direct cost of an amputation, just the direct cost, not the indirect cost, is $50,000. Right. So I do think there's a large market and a large opportunity. Again, we have to prove it, but the numbers in our view do add up to sort of signal something very, very significant.
And it qualifies as an orphan indication, did you say earlier?
Yeah. Yeah, you can. You can.
Yeah. And do investors actually get excited about those kind of things? Like I feel like there's the orphan designation and also the maybe fast track kind of or breakthrough statuses that you can get.
Right.
Are you able to get all of those for what you're doing?
So we got fast track and orphan in CF, and I think DFO is now next because we now have the data. So that's definitely in the cards, and you're totally right. And I think that's something really smart we heard from our investors is, look, you're a biotech company, right? You got to be smart in the way you develop, and you want to choose, orphan indication for its benefits just because it's so hard. Just trying to do everything you can to shorten development timeline, reduce the cost of the studies, reduce the number of patients, etc.
Yeah. And the shifts, I also wanted to touch on the FDA. Obviously, there's so many shifts happening over there. First, I want to know, like, is that impacting you now? And then on the positive side, I saw something out that FDA had said that they're not going to require animal studies as much. Does that factor into things, or I think the phage field has already kind of benefited from being able to
Right.
skip some of that already. So,
Right.
yeah.
I think again, I got to say, our interaction with the regulatory agencies were very favorable, right? So I hope nothing changes because they were fantastic. I think we didn't need to do animal, at least the FDA in 3 public workshops said there's no need to do animal studies and no need to do healthy volunteers. So I think we've gotten a lot of support from the agency, very open-minded. I think we're gearing up to discuss with them real world evidence and sort of exploring microbiology endpoints for our CF study. And that's a study that will take place later this year. So again, I don't know. We don't have perspective of the changes right now, but I must say that the agency to this date has been super supportive, open. Discussions were also very, very scientific. There's a lot of consideration. I think you spoke in some of the earlier podcasts about manufacturing and like whether it causes generalized transduction and harmful genes. And again, everything was to the point, practical, and we just made the dynamics were just very, very good. So let's just hope it stays the same. If it improves, even better, right? But at a minimum, let's hope it stays the same.
So, if I could ask a very specific question regarding the FDA, and something I've always been curious about for a while, is that you mentioned, I think you have around 50 staph phages, and that you're sort of screening among those phages for the phage that's going to be used in the DFO study for an individual patient. And I've always been curious about that because I guess it's a little bit like CAR T treatment for cancer in the sense that I could imagine a scenario where like you conduct a successful clinical trial and you only use 20 of the 50 phages that you have. And then you're presented with this possibility that you could get an indication. And then the first patient that's used post the indication, phage that's one of the 30 that wasn't involved in the study, is the phage that you decide you select. So in that sense, it would be the process of how you pick the phages and how you pick that phage for that patient, rather than the fact that that phage has been in the clinic at all. How have you had broad conversations or specific conversations with the FDA or other regulatory agencies around that?
Not at that level. I mean, you got to remember, I think that's sort of like what, again, right, it kind of goes to our conversation like worrying and doing the experiment. Think about it, even just with standard antibiotics, like their coverage in strains is like 90%, 85%, right? So even standard antibiotics don't cover all the patients, right? And they don't test for sensitivity either, just do the clinical study. So I think it's one of the things we worry about. What are the next steps in DFO, right? Obviously, I'm optimistic there'll be less of an issue than we think. That's what we've seen in CF, and to some extent also in DFO, right? Like in CF, we put together the cocktail. Again, we did make sure we tested, we made sure that the sensitivity, but then it opened up this discussion like, wait a minute, are we culturing all the strains there? Maybe there's other strains, maybe different parts of the lung might have like different strains, right? And do we really know the full picture? And we don't know, right? And then you kind of say, okay, let's just do the experiment. So I suspect, you know how it is. We'll have to choose the bank of phage that we'll produce, whether it's a cocktail or sort of the phage that we'll take forward, and we'll have to see how the clinical study looks. But again, in Staph aureus, I think it's less of an issue, just based on the data we've generated to date. Right? If this was Mycobacterium, then I think we'd be really worried. Right? That's a really tough one.
I think we should probably wrap up because we're at the top of the hour. But I have a couple of rapid fire questions left.
Go for it.
So, okay. One is, I forgot to ask about quantity. How much phage do you need to get to the site, or that you need to dose, or how much did you use in this study?
Yeah, we haven't disclosed it. I can say that the vials were not humongous, right? So, and the titers were not like extreme. We didn't sort of optimize super high titers. So again, it seems that there's enough phage to start probably the amplification process, and it just worked, right? And again, that's what was used in compassionate use cases either, not as well.
And then what, if you had to say, since our audience is very much going to be phage researchers, what research is left to do that would be the most impactful for you? Like if you saw a paper published tomorrow, finally answering some question about staph phage biology, or even maybe on the pharmacokinetic side, what are you thinking?
So I think there's a lot of great work happening right now in defense systems. So that field is sort of like moving. The field that has sort of surprised me that there isn't that much work is actually biofilm. Right? I think there's a lot to learn about biofilm. In DFL, you can literally image the biofilm. And, we use our assays for biofilm, but I suspect that there's so much more work that we can do in biofilm to harmonize assays and understand. By the way, sometimes biofilm could be polymicrobial, right? So what does it mean? And we know that the bacteria produce different biofilms in different conditions. So I think that's one of the field that we're surprised that there isn't a lot more work, because I suspect that that's one of the leading causes for failure of standard therapy, right? Of antibiotics.
Right. Okay. So researchers, please study the biofilm formation.
And let us know. Reach out to us because we're sweating.
Okay. Yeah, maybe would you collaborate with academics if they had a new assay for biofilm or something like that?
All the time, as much as we can.
Awesome. The final kind of question that we ask everyone is about where you hope and expect BiomX to be in the future. So the 5-year and the 15-year.
So we had this discussion already internally in the company 5 years ago, and wanted to be now with like 2 programs in phase 2, and somehow we got there. So I think we're very fortunate. Again, it's thanks to the hard work of the BiomX team and the Adaptive Phage team. In 5 years from now, hopefully we have like 1 or 2 approved drugs. In 15 years, we want to be sort of a go-to modality, right? We want to have multiple programs that we address. I mean, I'm sure some of the cases of infectious will be retreated with small molecule antibiotics because it's better, but I hope that there'll be more and more indication that we can pursue. And also, I think going back to where we started, more chronic disease, such as inflammatory bowel disease and some of the others, which are really much tougher. Once we feel comfortable with the modality, we can start exploring the target.
Some of the ones that BiomX started before you guys merged with APT.
Right. Before we put it on ice and focused on infectious disease, I think we'd love to kind of revisit that, but later.
Yep. Awesome. Anything else, Joe?
No, as usual, I could ask questions forever, but I think in the interest of time, this has been great. We hit a lot of important issues, and thank you for your thoughtful responses and taking the time to talk to us. We always appreciate that.
No, it's a pleasure, and keep up the great work. Hopefully we want more people in the field. So we're at your service to help in anything, and keep up the great stuff you're doing. The field needs it.
Thank you. Thank you so much for being here. This is so much fun. See you around.